Hello, am I audible? Okay, so it seems that students are still joining. So we will wait for 2 to 3 minutes. Let everybody join and then we'll start the session. Okay. Hello. Okay, so I think we'll start the session, and as students will join, we'll just let them in. Okay. So I'm going to share my screen. Um, okay. So let me know, is my screen visible or not? Fine. So is my screen visible?
Yes, ma'am.
Yes, ma'am. Okay. Students are still joining. Fine. So, a very warm welcome, everybody. So, I am Apurva Soni. So, I am a PhD scholar. I am a Prime Minister Research Fellow, and I am working as a teaching assistant for the course Genetic Engineering Theory and Application. Okay. So, every week we will have this live class. So, in the live class, what we will do is that we'll try to deal the questions of the topic that we have learned in the previous week. Okay, so this time, since 2 weeks have already been completed, week 1 and week 2, so we will do the questions related to the content which was covered in week 1 plus in week 2. Okay, so just a minute. Okay, still people are joining. Hmm. So in the first week we have learned about the basics of biological system. Okay, so our lecture 1 and 2 and 3, they were related to the cellular structure and the basics of biotechnology. So in lecture 1 we have studied about what is the basic definition of biotechnology. Okay, so what is biotechnology? Biotechnology is nothing but the summation of technological tools and living organisms. So basically, with whatever knowledge that we have about life science of biology, okay, now we are using some technological aspects into it so that it can be beneficial for the humanity, it can be beneficial for the society. Now comes the point that what kind of benefits does biotechnology give us? So, it can be the improvement of the livestock, the purity of the livestock. Then, with the help of biotechnological applications, we are able, and like in the COVID scenario, also, everybody saw that it is biotechnology only which helps us to make new vaccines, to develop new drugs to combat various diseases. Okay, so biotechnology is very much helpful in medical sciences. Also, biotechnology is very helpful in agriculture sciences, and there are vast variety of applications of biotechnology. So there is essentially a very much need of biotechnology. Then we have studied what are the important milestones of biotechnology, then the applications of biotechnology. So as I mentioned, some of the applications, biotechnology is very much important in agriculture, pisciculture, poultry, vaccines, medicine, genetically modified organisms, drug delivery. Then we have discussed about the genetically modified organisms. That means the basic strategy of developing a genetically modified organism, right? So, for developing a genetically modified organism, there are two things that are needed. First is the, can anybody tell me what are the two things that are required for any development of genetically modified organism? Anybody who remembers from the class? Anybody who wants to try? Anyone?
Genome and plasmid.
Genome and plasmid. Okay. So we required the genome, that means the exact DNA that we want to insert into our plasmid, correct? And then we want a host. Now, when we talk about host can be of prokaryotic host and it can be a eukaryotic host also. So, first we will see about the basic. This is something which is very basic. We have all studied what are the properties of prokaryotes, and then what is the difference between a prokaryote and a eukaryote. And then we have studied about the basic gram staining method, that is, how to stain the gram negative and the gram positive bacteria. Okay. So, what are the basic properties of a prokaryote and how prokaryotes are different from eukaryotes? Just three differences. Anybody who wants to give a try? Basic difference, very basic. The first thing what comes into our mind when we think about a prokaryote and a eukaryote? First thing, what comes to our mind when I say to you, when I ask you all that what is the difference between a prokaryotic cell and a eukaryotic cell? What you can think? What is the difference?
Compartmentalization.
Compartmentalization. So what I'm trying to say here is nuclear arrangement, okay, but the basic, the first thing that comes to our mind when we think about the difference of prokaryotes and eukaryotes is that
Single cell and multiple cells.
prokaryotes do not have well defined organelles, but eukaryotes, they have well defined organelles. There are no organelles which are present in prokaryotes. Even the nucleus is not in a particular even the nucleus is not there, it is just a nucleoid material is there. So the first basic difference that comes to our mind when we think about a prokaryotic and a eukaryotic cell is that prokaryotes do not have well defined organelles, whereas eukaryotes, they do have. Okay. Now, uh, prokaryotic cell can be of two types. It can be a gram positive or it can be a gram negative cell. Now, what is a gram positive and a gram negative cell? So gram positive and gram negative depends upon the peptidoglycan layer which is present. Okay. So gram positive have a thick peptidoglycan, whereas gram negative have a thin peptidoglycan, correct? Now we'll come to the first question. The first question is, movement of a bacterial cell is facilitated by, what can be the answer? How the
Flagella.
flagella, correct. So movement of a
Flagella.
bacterial cell is facilitated by flagella, correct. Now what is the function of capsule if it is not helping in this thing, locomotion? Then how capsule is helpful? What is the function of capsule?
Protect bacteria.
So
Outer cover
capsule
of
is,
the layer.
it is protection. So capsule is required for protection. Flagella is required for locomotion. Plasmid is, what is plasmid?
extra genome.
extra circular DNA, extra DNA, which is present in the eukaryotes. Okay. Now, next question is, which of the following is a true statement about eukaryotes? The first question is, eukaryotes have introns. B, eukaryotes have 70s ribosomes. C, transcription and translation are coupled in eukaryotes. And D, eukaryotes have single origin of replication. So, what is the answer?
Presence presence of intron.
Presence of introns. So, eukaryotes do not have 70s ribosomes because they have
80s ribosome.
80s ribosome. And transcription and translation are not coupled in eukaryotes. Why they are not coupled? Because transcription occurs. Where does the transcription occurs and where does translation occurs in eukaryotes?
Nuclear transcription and translation occurs inside.
So in prokaryotic cells, since there are no well-defined organelles, so that's why both transcription and translation are occurring simultaneously in the same space, okay? Whereas it does not happens in the case of eukaryotes. And eukaryotes have multiple origin of replication. Now, third question is, for producing a genetically modified organism, the gene of interest is cut with a restriction enzyme named EcoR1. Okay. So, if this is our gene of interest and it is cut with a restriction enzyme EcoR1. Now, uh, now we have to digest the vector. So, for the digestion of vector, which enzyme should be used? Options are ZO1, SAL1, EcoR1, and none of the above. Which enzyme should be used?
R1.
EcoR1.
Eco
Why?
Eco R1
Why EcoR1 should be used?
because ligation required same cloning site,
This
cut
is
site.
the gene of interest, okay? And this is our vector. Here we are making a cut and here also we are making a cut. Now, if we want this insert to go here in the vector, then the insert and the vector should be cut with the same restriction enzyme. Only then it will be able to come and bind to this. Okay. So, if we have cut our insert with EcoR1, then we have to cut our vector also with EcoR1. Now, one important thing to understand over here is that here I have mentioned in the question just one this thing enzyme which is EcoR1, but you can cut your insert with two enzymes also. So you can cut this side with one restriction enzyme and you can cut this side with another restriction enzyme. So it is not that you just have to use one restriction enzyme, you can use two restriction enzymes also. So if you are using two restriction enzyme, the benefit that you will get is that you will know the orientation of the insert that you have added. What I mean to say is that suppose this restriction enzyme is here, and if this your insert, if this is going here in this direction only, you know, because this restriction enzyme site and this restriction site are here, so this end will ligate this side and this end will ligate this side. So suppose if here it is 5 prime and here it is 3 prime, so you will be able to know the orientation also. Okay. So if you are using double digestion, then it is better because it helps to know the orientation also. Next question is, identify the correct statement regarding the gram negative bacteria. So options are: A, gram negative bacterial cell has a thin peptidoglycan layer and lipoic acid. Gram negative bacterial cell has a thin peptidoglycan layer, outer membrane, lipoproteins. Gram negative has a thick peptidoglycan layer, outer membrane, and lipoproteins. Gram negative cell has cell stains blue during gram staining as they have thick peptidoglycan layer. So, what is the correct answer? What should be the correct answer here?
Option B.
Option B. It has a thin peptidoglycan layer and outer membrane and lipoproteins. So what is the basic difference between a gram positive and a gram negative bacteria? That gram positive bacteria, they have a thick peptidoglycan layer, whereas gram negative bacteria, they have a thin peptidoglycan layer. Apart from that, gram positive bacteria, they have lipotoic acid, whereas gram negative bacteria, they don't have lipotoic acids. And gram negative bacteria, they have outer membrane also, and they have lipoproteins also. So these are the basic difference between gram positive and gram negative bacteria. And on the basis of this only, your gram staining occurs. That okay, that we'll see in the next question. So during gram staining, the gram negative stains which color and gram positive stains with which color? Give me the answer.
Stain with pink color.
No.
Blue and
Gram
Pink
pink.
and blue.
gram negative stains
And
Blue and
blue
pink.
color.
first option.
No, it is not the first option. Gram negative stains blue color and gram positive stains pink color. So if we just look at the
Pink.
basic step, so what is the first step? What are the two dyes that we use? Safranin and
Crystal violet.
hmm. So first we
Crystal
will
violet.
stain. So first we will stain our cells with crystal violet and safranin. Then we will wash it with the help of non-polar solvents, which will act as a decolorizing agent. And then if the after the counter staining, the gram negative bacteria, it will give us a blue color, whereas a gram positive bacteria, it will give us a pink color. Okay, so then we come to the other sets of questions. So in lecture 2 we have studied about the we have continued about the host cell. And in host cell, in the week 1 we have studied about that host cell can be prokaryotic in nature. So what are the properties of prokaryotic host cell? What are the different types of gram negative and gram positive prokaryotes that we have studied? Now from prokaryotes, now we will shift to eukaryotes. So then we have studied in our second class that eukaryotic cells can also be host for our transgenic models for recombinant DNA and genetic engineering applications. But eukaryotic cells can be animal cells also and plant cells also. But animal cell differs from plant cell. The basic difference is that plant cell, it has a cell wall, whereas animal cells, it does not have a cell wall. Then plant cell, it has large vacuoles, whereas animal cells does not have large vacuoles. Then does animal cells have chloroplast? No. Plant cells, they do have chloroplast. Then plant cells also have glyoxysomes. What is what is the function of glyoxysomes? So glyoxysomes are used for the degradation of lipids. Okay, in animal cells we have peroxisomes, in plant cells we have glyoxysomes. Then we have studied about the various components or the various organelles of a eukaryotic cell. So in eukaryotic cell we have cytosol, nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, lysosomes, plasma membrane. Okay, so and now what is the difference between cytosol and cytoplasm? So cytosol is just the fluid part, whereas cytoplasm is the fluid part plus the various organelles which are present. Okay, am I saying correct or not? Tell me,
Yes, ma'am. Yes, right,
am
ma'am.
I completely saying correct or is there anything more which should be added here? If I talk about cytoplasm, it is the fluid part, okay, plus various organelles. Okay, anything else? Very important. Cytoplasm, when I talk about, eh, one thing that I have to exclude is nucleus. That means cytoplasm is fluid plus all the organelles except the nucleus. Okay, that is important to know. So cytosol is just the fluid part, cytoplasm is fluid plus organelles except the nucleus. Except the nucleus. Then we have studied about mitochondria and ATP synthesis. So in the lecture there was this thing: open conformation, tight conformation, loose conformation. So could you understand that topic, that part? Open, loose and tight conformation. Just tell me. Anybody?
No,
Ma'am,
ma'am.
can
Can you
you
tell?
just revise it?
Okay, so
Yeah.
that's why I was asking, because I thought that you would not be able to. So, here we are talking about ATP synthesis. Okay. So, ATP synthesis, we have an enzyme which is known as ATP synthase. Okay. So, just look at this ATP synthase. Students are still coming in. It's creating problem in between. Okay, so just look at this figure of ATP synthase. Okay, so this ATP synthase has two subunits. The first subunit, it is the F0 subunit, and the second subunit is the F1 subunit. Correct? Now this F1 subunit, it is embedded in the matrix, whereas F0 subunit, it is in the membrane. Okay, so it is in the inner membrane, it is in the matrix. Now the F F0 and F1 subunit, how it works is that it works on the basis of the proton gradient. Okay, so it works, the ATP synthesis works on the basis of proton gradient, which is also known as PTN, that is proton motive force. So uh, I'll just copy this diagram so it becomes easier for me to explain. Right. Students are still coming. Fine. Now you see what happens is that outside there is a higher concentration of oxygen, hydrogen, and inside there is a lesser concentration of hydrogen. So always things which are at the higher concentration, they try to come towards the lower concentration. Okay, so because of this concentration gradient, this hydrogen ions, these protons, they try to come inside, and because of this ATP synthase, it rotates and then it produces ATP. So how it rotates and how it does is that ATP synthase, then it has the alpha beta subunits which are present in the ATP synthase. They are alternating, and there are three conformations: the closed, the open, and tight. So suppose there is an open conformation. First we'll deal with the open conformation. Now it is open, so what will happen? It is open, so ADP and inorganic phosphate, it can come because the gates are open and it is allowing the ADP and PI to come inside. Okay, now ADP and PI has come. Now what happens is, or let me call this as mm, loose. Hmm. This is loose. Now what happens? ADP and PI has come. Now comes our loose conformation. So what happens? That from open, now it will come and it will come a little bit close. It is still not completely together with each other. But if suppose this is ADP and this is PI, so in loose conformation, they come a little bit close. And then in tight conformation, they are so tightly packed that now ADP reacts with PI and it forms ATP. Okay. So this is how the three conformation works. In open conformation, what happens? In open conformation, in open conformation, ADP and PI, it comes. And in tight conformation, actually ATP is formed. So, in regard with this, first we'll solve this question and then we'll go to the another question. It's a very simple question. Just now I explained to you that the F0 and F1 subunit, it is present in F naught is present in inner where it is present till F naught is present in
Inner membrane.
and F1 is present in.
Embedded with cell membrane.
Okay. Now another important point is
Which is
that
embedded, ma'am.
if you can see that this is the F naught. Now so ATP synthase has two subunits, F naught and F1. And this F naught and F1 again they have many subunits. They have again many components. So F naught has A, B and C, whereas F1 has a lot many subunits such as alpha, beta, gamma, delta, epsilon. So these all are present, and the three conformations that I was talking about, they are actually the conformations of the beta subunit. Okay, so it is like alpha beta in a round circle it is present, and in the between there is gamma subunit. Then we'll solve the next question. The transcriptionally inactive part of the nucleus is, what is the answer? Very simple.
Heterochromatin.
Okay. Second is, in mitochondria the inner membrane is folded into structures which are known as
Christy.
cristae.
Crystal.
And
History.
in chloroplast, what it is called as?
Cistern.
Yes, good. And then what are this thing granum and what are thylakoids?
Thylakoids are present
Thylakoids are
in chloroplast.
sacs of granum.
So thylakoids, they stack with each other to form granum. Okay. Then this question we have already solved. Then organelle which is used to track the tree of a family. Which organelle is used for family tree tracking?
Mitochondria.
Mitochondria. Can anybody explain me the reason behind it? Why mitochondria is used? Okay. So see,
Mitochondria
huh,
Because
carries the
ma-
gene of the parent plant.
see,
Especially
during
nucleus, which carries the nucleus.
huh, yes, correct. So basically what happens that during fertilization, this is the ovum and this is the sperm. Okay. So during fertilization, sperm, it loses its nucleus. Okay. But the nucleus of the ovum is still there. And mitochondria are present in the nucleus. So that's why the mitochondria from the mother genome, it is always transferred from generation to generations. So that is why mitochondria can be used for tracking the family tree, and therefore it is very much importance when we consider the maternal inheritance also. Mitochondria is basically related to maternal inheritance because mitochondria is present in the nucleus of the mother. Okay, because the nucleus of the sperm is lost during fertilization. So that is the reason why mitochondria is used for tracking the family tree. Okay, next question is that the light and the dark reactions in chloroplast, it occurs in where the light reactions occurs. Light reactions occurs in thylakoid, and dark reactions, they
Thylakoid and
occur in,
stroma respectively.
huh, this is the answer. Okay, so next question is, the vesicular traffic system comprises of, so before this what is vesicular traffic, just I'll just quickly recap it. So when we talk about endoplasmic reticulum, there are two kinds of endoplasmic reticulum. The first is rough endoplasmic reticulum and the second one is smooth endoplasmic reticulum. So rough endoplasmic reticulum is known as rough because it is attached with the ribosomes. Okay, and ribosomes are the site of protein synthesis. We all know this. Now what happens that when a protein is synthesized in the ribosome, then it goes immediately to the endoplasmic reticulum. And which endoplasmic reticulum? Rough endoplasmic reticulum. Now what happens that when a protein is synthesized, it is very naive. It requires a lot of modifications. So in rough endoplasmic reticulum, basically this protein is modified. Okay, this protein is modified. What kind of modifications? N-linked glycosylations and other many other modifications it occurs in the rough endoplasmic reticulum. Now rough endoplasmic reticulum is saying that okay, I have done my job. uh, Ribosome has given me the newly synthesized protein. I have made it properly. Now just a few modifications are left, and also I this protein needs to know where it has to go, like whether this protein needs to go to the mitochondria or this protein needs to the chloroplast, but that is not my job. Then comes into play your Golgi bodies. Golgi body says that okay, you give me the protein, I will do any other modifications that are left, such as O-linked glycosylation, and I will also tell these proteins that where you have to grow, where you have to go. So what I will do is that I will do the tagging. Okay, so I will tag the proteins so that now the proteins know where they have to go. So first the proteins are synthesized in the ribosomes. Then in the ribosomes, the newly synthesized proteins, they go to the rough endoplasmic reticulum. Rough endoplasmic reticulum, it modifies the proteins and then it sends to the Golgi bodies. In the Golgi bodies, some other further modifications which are left, that happens, and then it tags the protein so that the protein knows where it has to go. And here we are saying that vesicular trafficking may we are okay, this much I'll tell. So then what would be the last organelle where it will go according to the vesicular trafficking system? After endoplasmic reticulum and Golgi bodies, this I've already told you. What would be the
Lysosomes.
answer? Lysosomes. So that means now this is basically vesicular trafficking, so that now the protein that has been synthesized, it will be packed and it will be transferred to its respective place. Okay, so now this rough endoplasmic reticulum has a lot of functions. Then what is the importance of smooth endoplasmic reticulum? Then what function does smooth endoplasmic reticulum will do? Because it is not involved in vesicular trafficking, but it will do the kind of detoxification. Okay, it is involved in detoxification. Then it is also involved in the synthesis of hormones. So smooth endoplasmic reticulum, it is involved in synthesis of hormones and detoxification. Okay, is it clear to everybody, the vesicular trafficking system? Now we'll
Yes,
come to
ma'am.
the okay,
Yeah.
good. Now we'll come to the next question. Matching, it's very simple. So Golgi bodies, what is the function of Golgi bodies? Just now I told
Protein
you
sorting,
protein salting. Lysosomes,
degradation of
Degradation.
pathogenic bacteria.
Bacteria.
okay. Rough endoplasmic reticulum,
Steroid hormones.
no,
Sorry,
no,
No.
sorry.
no.
Protein
Ribosomes.
synthesis.
Ribosome,
Ribosomes.
See, rough endoplasmic reticulum is related to protein synthesis, and protein synthesis is related to ribosomes. So in this type of matching, you can just you have to try to correlate. If A is equals to B and B is equals to C, then A can be equal to C. In these types of matchings, then smooth endoplasmic reticulum is steroid hormones. Okay. So now we'll come to the hmm, then we'll come to the last lecture of week 1. So in the last lecture of week 1, we have studied that okay, now we know the basic properties of a prokaryotic cell. We know the basic properties of eukaryotic cell. We know what is the difference between a prokaryotic cell and eukaryotic cell. Now suppose I have to isolate different organelles or different fractions of a prokaryotic cell or a eukaryotic cell. How can we do that? So that we have studied in our lecture 3. So first we have studied the fractionation of a prokaryotic cells. So in a prokaryotic cell, since there are no organelles, so this is not much that we can isolate. What we can isolate is the cytoplasm and the periplasm. And the reason for isolating these two is because these two are the regions where from which we can get proteins. Okay. So periplasm you can do with the help of, for periplasm we can use the technique of osmotic
isolation,
shock. And for cytoplasm, what we can use? Sonication. Okay. Now it has a the protocol that you have all read in the lecture. That is a general protocol. Now this protocol, the basic thing that you have to understand is that whenever you are taking all the components, you have to centrifuge it. Then according to the sedimentation coefficient, the density of that particular organelle, you will harvest that particular supernatant. Okay, so that depends. The exact protocol is not that much important, but the logic and the concept behind using that protocol is very much important. Because if anybody of you want to go for further research in PhDs, then what is important is not that what you are doing, the technique is not important, the logic behind that technique is very much important. Because doing a PCR, setting up a PCR reaction, is something that what a technician can also do. But what separates a technician from a researcher is that a researcher knows the logic behind it. So if you want to go for higher studies, if you want to go for, if you are in undergraduate level, if you go for masters, the master students who wants to pursue PhD, important thing is to understand the logic behind any protocol that we are using. Okay, are there any master student here in this course, or all are graduate student,
Yes, ma'am.
bachelor student? You all are bachelor student? Okay, so there are some students. So you will have a 6-month project also, right? Will you have a project also?
Yes,
The
ma'am. In
bachelor
fourth
students.
semester.
In fourth semester you will have a project. So whenever you are going for a project, whenever you're going for any kind of research or any kind of training, it is important to know the logic and the concept behind the technique that you are using. If you are using NaCl, why you are using NaCl for DNA isolation, why you are doing this. So the logic is important. Anyways, then we have studied about the fractionation of a eukaryotic cell. So in eukaryotic cell, the important organelles that are routinely fractionated for various experimental procedures, it is mitochondria, plasma membrane, chloroplast, cytosol. And basically there are two types of methods or techniques that can be used. The first one is the differential centrifugation and second one is the density gradient centrifugation. So when we talk about differential centrifugation, it depends on the sedimentation rate. Okay, and density gradient depends upon the density. So sedimentation rate means that suppose if uh, if you are centrifuging any cell, then the heavier particles will settle down first, then the lighter will be above that. So the sedimentation rate will discriminate the different organelles which are present inside the cell. And density gradient means that you have this particular tube and you have used a buffer, and this buffer will make different densities. Okay, this buffer will create different densities. Now your substance X, A, suppose your substance A that you want to isolate, and this is the density number 1, 2 and 3. It has a density of 3. So when A, the density of A becomes equivalent to density of 3, then the A will appear here. A band will be observed here. And then we can just pick this band. So when the density of our organelle corresponds to the density which we have created in a tube, then a band will appear and we have to take this band with the help of pipette or some collectors. Next, now we'll be doing the questions of this particular chapter and we have to move a little bit faster, I think, because there is a lot left. Anyways, the function of TAC buffer during fractionation of a prokaryotic cell is what this buffer will do? Tell me. Anybody?
Provide provide density.
No. This even an option? No, that is not even an option. So suppose these kinds of questions comes for if you're going to give any gate or something. Do you know the full form here? I know nothing about this question, but I think this s something it has to do for sucrose, I'm assuming it. Okay, and now I know that sucrose is basically required for giving the osmotic shock to the cells. Okay, so basically, I just told you that for prokaryotic cells, we can use osmotic shock as a technique for the isolation of periplasm. So, for providing the osmotic shock, sucrose is used. Okay, so basically, this TSC buffer has sucrose, and this sucrose helps in providing osmotic shock. So the answer would be A, to provide the osmotic shock with the sucrose present in the buffer. Okay. Then sonication and osmotic shock strategies are applied for what? Just now we have studied, just
Option B.
discussed sonication for cytosol, osmotic shock for periplasm. Yes. Good. Next question. Which of the two properties are exploited for fractionation of organelles? Again, we have just discussed that for the fractionation of organelles, we require the sedimentation rate and density. Okay. For, sorry, were you saying something? Anybody was saying something?
Ma'am, I am saying that option B is correct.
Option B is correct. So for sedimentation rate, we can use differential centrifugation. For density, we can use the density based centrifugation. Now in density based centrifugation, band is observed when the band is observed. Just now we have discussed when the band is observed.
Ma'am, first option, density of the media is equal to the density of sample.
Yes, good. So just now that we have studied that in density based centrifugation, okay, suppose this is our tube and we have filled a buffer such a way that this buffer has different densities. This is density 1, this is density 2, this is density 3. We have poured whatever solution we have and then we'll centrifuge it. Now what will happen? This we want to isolate something, an organelle X, and this X corresponds to the density 2. So what will happen? So when X reaches its corresponding density, it will give a band at 2. Okay. So you are it is centrifuged, and the density of X, when it corresponds to the density which is present at 2, then there will be a band observed that will stop moving. And now with the help of pipette, we'll just pick this band. We'll pick the band. Okay. So density gradient centrifugation, density of the media should be equal to the density of the sample. Okay, so this was about the module 1. So before coming to the module 2, that is the course that we have discussed in week 1. So what all we have studied in week 1? We studied that what is biotechnology, what is the need and applications of biotechnology. Okay, just have a quick recap of what we have studied in week 1. Hmm. We have studied about what is the definition of biotechnology, its need, its applications. We have studied about the basic strategy for creating a genetically modified organism. For this, we need many kinds of hosts, such as prokaryotic host and eukaryotic host. Then we studied the properties. We have studied about gram positive, gram negative. In eukaryotes, again, we have studied about the different components. Okay. And then ultimately we have studied about the isolation of different components. Okay. So this we have studied. So do you have any confusion in week 1? You can just ask me before we switch to the week 2 module. If you have any doubts, you can ask me now. Any doubts? Anything that you could not understand from the lectures, or by the any question that you have any doubt, you can just ask me. Should I
I don't understand the part of gram negative and gram positive.
can you repeat? You want to know the difference between the gram negative and gram positive bacteria, am I right?
Yeah, yeah.
Okay. So
Um, Color staining.
color staining, gram staining, that you want to know. Fine. Okay. So basically, the gram staining works on the basis of the peptidoglycan layer. If the peptidoglycan layer is thick, it is able to take the stain. Okay. And that is why when we add the decolorizer, we still get the pink color. But if the peptidoglycan is very thin, then when we add the decolorizer means add any kind of non-polar solvent, then most of the stain is washed away and we get a blue color. Now,
No, just reverse, I think. Because
I am
crystal
talking
violet,
about gram negative.
Ma'am,
Yes.
sometimes while doing gram positive, it's okay if we don't add decolorizer, right?
Okay. So,
No, like you wrote decolorizer, so it's in most of the experiments we don't use decolorizer.
will you try to then how you do it? Then how you are able to this thing uh, uh, identify the gram and the gram negative and gram positive bacteria?
No, because last semester we had a practical where we have to identify the same gram negative and gram positive. So while doing the gram negative one, we used I got it wrong.
No, if you have any, someone else was also saying something.
No, I had this
Ma'am,
question. I had
hello.
this question.
Okay, first I'll explain the difference between gram negative and gram positive. One student has asked, sorry, gram negative and gram positive bacteria. So first I'll explain that and then we'll come to staining. I think one student has asked about that. I forgot. Yeah. So gram positive bacteria, it has a thick
peptidoglycan layer.
peptidoglycan
Peptidoglycan layer.
and it has a thin
Peptidoglycan layer.
Outer membrane, no outer membrane, not thin outer membrane.
okay. Okay. So here outer membrane is present or absent?
Absent, ma'am.
In gram negative, outer membrane is present or absent? Think and answer.
Outer membrane is present, thin peptidoglycan layer in gram negative bacteria.
Hmm. So outer membrane is present in gram negative bacteria, and we have lipoproteins also. Here we have what we have? Lipotoic acids. In gram positive, what we have?
Lipotoic
Lipotoic
acid.
acids. Whereas in gram negative, we don't have lipotoic acids. And also, uh, which gram positive or gram negative, which bacteria can be used as toxins, and because of what? Lipid A is toxic. Okay. Now we'll come to the gram staining. So in gram staining, what we do is, the first step of gram staining is, many people have performed. Will you tell me the first step? What is the first step of gram staining? First we add
Staining with
crystal.
crystal
First we add crystal
crystal.
violet. Okay. Then what we do? Then we add a grams iodine solution.
Yes.
So
Yes,
this
ma'am.
grams iodine solution, what will it do? It will form a complex. It forms a complex with the crystal violet and iodine. Okay. Then what we do? We add a decolorizer. And then this decolorizer, what it does, it dehydrates
Decolorize the gram-negative bacteria.
the peptidoglycan layer. Okay. It dehydrates the peptidoglycan layer. So the peptidoglycan
Ma'am,
layer,
alcohol can be the decolorizer.
yes, alcohol can be a decolorizer, because decolorizer has to be a non-polar solvent, and alcohol is a non-polar solvent. Then it kinds of decolorize it. Then what will happen? Anybody?
We use saffronin.
No,
Saffronin.
this, see, now because the
Gram-negative.
peptidoglycan layer is shrinked, then this crystal violet, the complex, it is not able to penetrate, and therefore it is trapped in the gram positive. Okay. And whereas in gram negative, because the peptidoglycan layer is very thin, so this stain is not trapped, and therefore when we add a counter stain such as saffronin, so uh, uh, it in gram positive, it is able to disrupt the crystal violet, and therefore it gives a blue color, whereas in gram negative it cannot give. So the basic concept
Yes.
is that because of the peptidoglycan layer thickness, after adding the stain, it is getting dehydrated, which is causing the shrinking or the tightening, and therefore the stain, it is not able to wash off. Whereas in the case of gram negative, when the peptidoglycan is very thin, so thus dehydration is less. Okay. Now I'll come to the second this thing lecture. Otherwise, we'll not be able to finish. Okay. So in week 2, we have studied about the basics of biological systems. So now that we have studied about the host, prokaryotes and eukaryotes and its properties, our second objective was to know that what are the metabolic reactions that occurs inside the host cells. Okay. So when we talk about cellular metabolism, so cellular metabolism can be of two types. The first one is the catabolism, and the second one is the anabolism. So catabolism is the breakdown of the constituents, whereas anabolism is the synthesis of the constituents. So both catabolism and anabolism, it together, it forms the cellular metabolism of our body also, and cellular metabolism of eukaryotes. Now when we talk about, then we have learned about the glycolysis pathway. So a very quick recap of the glycolysis pathway, and then we'll do the balance sheet here. We'll try to see how much ATP is formed. Okay. So in the first step, you can say the first initial steps, you can see there is A and there is B, and this picture I have taken from Behringer. So first is the preparatory phase, and then is a payoff phase. Preparatory phase means that first there is some input of energy, input of ATP is needed, and then when you give some initial input of energy in the form of ATP, then your pyruvate is generated. So first step is the glucose is converted into glucose 6 phosphate, and here one ATP is used. So simultaneously I'll use the used, यहाँ पे one ATP is used. Now glucose is converted to fructose 6 phosphate, and then fructose is converted into fructose 1,6 bisphosphate. Again there is one use of ATP. Okay. So in the first step and in the third step, we have used one ATP. Okay. Now we come here, you can see that glyceraldehyde 3 phosphate, when it is converted into 1,3 bisphosphoglycerate, then there is a release of 2 NADH. अब यहाँ पे I'll write the release. release of ATP. Okay. So there is how much? How much is there? NADH is there, and NADH is equals to how many ATP?
3. 3
3
NADH is
ATP.
equals to 3 ATPs. Then again there is ATP here. 1 ATP is there. Okay. Then again you can see in the last step, phosphoenol pyruvate is converted into pyruvate. Again there is a release of ATP. Okay. But how much it is coming? 3, 4, 5. But you see that one glucose is producing two pyruvate molecules. So actually there is a release of 2 NADH, 2 ATP, and here also 2 ATP. Okay. So it will be actually 6 plus 2 plus 2, which is equals to 10.
10.
Okay. So 10 ATPs are released and 2 ATPs are used. Therefore, net ATP would be 10 minus 2, which is 8. Okay. So in the glycolysis pathway, the net production of ATP is 8. Why? Because 2 ATPs here, this ATP and this ATP, they are used up. And then there are 2 NADH. There are 2 ATPs. Here also, here also. Okay. And 1 NADH will give us 3 ATPs. So likewise, in glycolysis, there is a net gain of 8 ATPs. Then we have studied about the regulation of glycolysis, the glucose mediated control covalent modification, allosteric modifications, etc. Now we'll do some questions. So how many water molecules are used in glycolysis? Just look at this figure. How many water molecules you can see?
3.
How three? Can you tell me where the 3 molecules are?
2 water molecules.
In which step you are saying? Then tell me the step also. In which step you can see the usage of water molecules?
From
The
2-phosphoglycerate to phosphoenol pyruvate to water releases.
okay. So answer is 2. Then glycolysis takes place in
Cytoplasm.
okay. And Krebs cycle takes place in
Mitochondria.
okay. Next question is net gain of ATP in glycolysis is
8. 8.
8 because the release is 10 and use is 2. Therefore, net is 8. Then which of the following is the first step of glycolysis? Breakdown of glucose, phosphorylation of glucose, conversion of glucose into fructose, or dehydrogenation of glucose? What is the first step of glycolysis?
Phosphorus.
Phosphorylation of glucose.
Phosphorylation of glucose.
Phosphorylation, because glucose is converted into glucose 6 phosphate. So it is a phosphorylation of glucose that occurs. Okay. Then next question is the number of molecules of pyruvic acid formed from one molecule of glucose at the end of glycolysis is 1, 2, 3,
2.
4.
3 pyruvic acid.
2 pyruvate molecules. Okay. Then during glycolysis, the coenzyme Mg2 plus is required at which step? Phosphorylation, isomerization, final synthesis, dehydration step. At which step it is required?
During phosphorylation.
During phosphorylation of glucose.
Okay, now one trick that I will tell you, that if you know the function of Mg2 plus, then you need not to even look at the options. See, Mg2 plus is basically a coenzyme which is always required with matlab kinase. Okay, so Mg2 plus is always involved during the phosphorylation events. So suppose if you don't know anything but you know that Mg2 plus is related to phosphorylation, then you can tick that okay, Mg2 plus is there, Mg2 plus is required with hexokinase matlab kinase enzyme, so that means it must be the phosphorylation. Okay, so if you know the basic functions of the cofactors which are involved, then you can try to guess the answers if you don't really know the pathway also. Now, next question is, which hormone is important for the regulation of glucose metabolism? Estrogen, epinephrine, insulin, and oxytocin.
Insulin.
Hmm. So, how this insulin is helping us? So, basically, what happens that insulin is there, okay? Now, it will bind to
Insulin is
its.
helping to convert glucose to glycogen in the liver cell.
Okay.
It
So, let me
helps to
explain.
break down of glucose.
Yes, it helps to break down the glucose. So if glucose, okay, first I'll explain, then I'll ask the question. So insulin is there. So what will happen? It will bind to its insulin receptors. Okay, hormone is there. So hormone for any downstream metabolism, if hormone is there, it has to bind to its receptor. Every hormone has a receptor, and then only we are able to then only that hormone is able to transduce its signal. So insulin, it binds to the insulin receptor, and then insulin mediated downstream signaling occurs, and then two glucose transporters which are known as GLUT3 and GLUT4, what does it do? It goes to the membrane. Okay, so first insulin has binded to its receptor, then downstream signaling has occurred, then these transporters GLUT3 and GLUT4, it goes to the membrane, and what happens, it allows the glucose to come inside. So this transporters, it allows the glucose to come inside. Now as the glucose has come inside, then the glycolysis will take place. So when insulin is present, insulin is helping in the glycolysis process, that we all know, because we all are aware diabetes is a very famous disease. Everybody is aware that insulin is problematic and there is some problem with insulin is not getting secreted, or insulin is not getting bind by the receptor, and therefore diabetes is happening. But what is actually happening, what actually happens is that when insulin binds to its receptor, the transporter, it goes to the membrane and it allows the glucose to come inside, and therefore glycolysis occurs. Okay, but if the glucose insulin level is very high, then what happens is that sorry, if insulin level is very low and glucose level is very high, then what happens? If glucose insulin levels are low, if insulin is low, then what will happen? Glycolysis will be high or low? Glycolysis will be low, because when insulin is binding to the receptor, then only the whole downstream signaling is happening, and then only this transporters are coming to the membrane, and actually these transporters are only allowing the glucose to come inside now. So if glucose is only not present, the first step is only missing, then all this cycle will not occur. And if this all this synthesis will not occur, therefore glycolysis will not take place, and because glycolysis will not take place, the glucose level is high. So in diabetes, because insulin is less, so glycolysis is inhibited. Okay, did you understood this?
Yes, ma'am.
So basically, diabetes, what happens? There are two conditions. First is that insulin is not present, and because insulin is not present, so the whole downstream signaling is not happening. Therefore, glucose is not getting converted to glycolysis. The second reason of diabetes is that insulin is present, but there is some problem in the insulin receptor. So this receptor is not able to recognize insulin. Insulin is there, but there is problem in the receptor, And because this receptor is not able to recognize insulin, the whole signaling is not occurring, and therefore glycolysis is not taking place. And because glycolysis is not taking place, glucose is not getting breakdown. And because glucose is not getting breakdown, glucose level gets high and you get diabetes. Now, the activity of the glycolysis pathway is reduced during, just now we have in detail we have discussed this right now. What would be the answer? High blood glucose level, high presence of ROS, presence of ATP, low blood glucose level. Activity of when glycolysis is not needed, or when the glycolysis is reduced.
When
High
the
blood
glucose
glucose
uptake is
level.
low.
Hmm. Okay. So now we'll study the next cycle, that is the Krebs cycle. Okay. So here I'll do the balance sheet for the Krebs cycle also. The first this thing NADH, it is not written over here, but now that we have synthesized pyruvate. Now pyruvate is synthesized into acetyl coenzyme A, and this acetyl coenzyme A can go to the mitochondria. Okay. Now what happens is that when acetyl this is the whole cycle. Now we'll see the balance sheet of it. So here the conversion of isocitrate to alpha ketoglutarate, 1 NADH is released. Okay. So I am writing here 1 NADH, which is equals to 3 ATPs. Here 1 NADH is released. Okay. This is the first decarboxylation reaction which is occurring. The first decarboxylation reaction that occurs is when isocitrate is converted into alpha ketoglutarate, then the first decarboxylation reaction is occurring. Decarboxylation reaction means decarbox matlab carbon dioxide is released. Then here the second decarboxylation reaction is occurring when alpha ketoglutarate is converted into succinyl coenzyme A, and again 1 NADH is released. Okay. So I will write 1 NADH, which is equals to 3 ATPs. Still students are joining. Fine. Then here succinyl coenzyme is converted into succinate. So here 1 GTP is released. So 1 GTP is equals to 1 ATP. So 1 ATP. Now succinate is converted into fumarate. Here FADH is released. Okay. So 1 FADH is released. 1 FADH is how many ATP? ATP? 2 ATPs. Okay. 1 FADH is equals to 2 ATPs. And again from malate to oxaloacetate, NADH is released. Now 1 NADH, which is equals to 3 ATPs. Am I missing something here? Can anybody tell me what I am missing? See how much it is coming. I'll tell you what I intentionally didn't write that to see whether you have really learned this pathway energy calculation or not. But it seems that you are still confused. Okay, so how much this is coming? It is coming 13, right?
13.
And there are, since there are two pyruvate molecules, so it should be 13 into 2, 26. But as we know that Krebs cycle, it produces 30 ATPs. That means there is, what is missing here? 1 NADH is missing. 1 NADH calculation is missing. So in the initial step, the formation of acetyl coenzyme A, 1 NADH is released. That I have not told you in the initial step only. So here it would be 1 NADH. which is equals to 3 ATPs. Okay, this is the cycle. But before this cycle, the production of acetyl coenzyme A, also one NADH is released, and that also we have to count. Okay, so what it will be then? Total, when you calculate it, it will come to how much? It will come to 15.
15.
15 ATPs. And since there were two pyruvate molecules which were formed by the glycolysis, so it would be 2. So which is equals to 30 ATPs.
30.
So that means in the Krebs cycle, how many ATPs are produced? 30.
30.
Okay. So how we have calculated? So one NADH is produced here in the production of acetyl coenzyme A. Okay. So here 3 ATPs, here 3 ATPs, here 3 ATP, here 1, here 2, here 3. Calculated 3, 3, 6, 7, 8, 9, 10, 11, 12, 13, 15 it is coming. Okay. And since there are two molecules of acetyl coenzyme A, so it will be 15 into 2, 30 ATPs. So in the Krebs cycle, how many ATPs are produced? 30 ATPs are produced. Is it clear to everybody?
Yes, ma'am.
Yes.
Okay,
Yes, ma'am.
so we have studied this balance sheet is very important. Okay, so if you are in your bachelor's, then throughout your this thing BSc or BTech, you will be having questions. You will be asked questions related to balance sheet. How many ATPs are produced? How many ATPs were used? How many ATPs were released? What is the net ATP? So these kinds of questions, they comes in GATE exams also. So this balance sheet is very important. Okay, and I have seen that students, they try to mug up this. They try to just mug and just do the rectification, that okay, I will just learn that 30 ATPs in Krebs cycle and 10 minus 2. They don't understand, they don't try to understand the logic behind it. So if you try to understand the logic and you try to see the pathway that is there now, then you would not need to mug up it. You will understand it and you will always remember it. Okay, so the only thing that you need to know is that at what places this NADH and GTP and FADH is released. One thing. Second thing, that one NADH will lead how many ATPs, one FADH will lead how many ATPs. These things you need to know. So it is very easy. One NADH means 3 ATP and FADH means 2 ATP and GTP is equals to ATP. So if you know these two things, then you will be able to calculate the energy calculations of any pathway. Okay, then we have studied about the regulation of Krebs cycle, significance of Krebs cycle, anaerobic oxidation, and significance of anaerobic oxidation. These are the things that we have studied in the first module. Okay, then we'll do some questions here. Dash accepts hydrogen from malate. Just look at the diagram. Dash accepts hydrogen from the malate. What accepts hydrogen from the malate? By looking at this figure, tell me. I think you guys have started sleeping now. 2 hours is a long time. Tell me, what is accepting hydrogen from malate? Oh God, it is in your screen. The answer is
NAD.
on the screen. It is in there. Okay, no worries.
NAD.
NAD.
NAD, correct. So see, here it is written that NADH is released. Okay, so NADH is released. What is the meaning of this? The meaning of this is that NAD, it has taken one hydrogen and it has produced NADH. Okay, FAD, it takes hydrogen and it produces FADH. So basically, if you see here, if I am saying that NADH is released or FADH2 is released or NADH is released, the meaning is that malate it during the conversion of malate to oxaloacetate, there is a removal of one hydrogen, and now this one hydrogen will not roam here and there. No, it will go and bind to something. So here in this case, the hydrogen has bound to the NADH. Here in this case, the hydrogen that was released, it has bound to the NADH. Here it has bound to the FADH. So what is the meaning of this release of NADH and FADH? This means that during the conversion of malate to oxaloacetate, or during the conversion of isocitrate to alpha ketoglutarate, or during this conversion, there is release of one hydrogen. Okay, so this release of one hydrogen, this hydrogen is taken up by NAD, and therefore it is converted into NADH. So anywhere when it is, where it is written FADH2 is released, that means earlier there was one FADH plus was there, which has taken one hydrogen and it is now reduced to FADH2. Okay, now FAD is reduced in which of the following reactions in Krebs cycle? FAD, where the reduction of FAD occurs? Here FAD is reduced
Succinate
where?
to fumarate.
Succinate to fumarate. So succinate to fumarate, the reduction of FAD occurs. Okay. Next question is, Krebs cycle begins with the creation of 6 carbon molecules as a result of a reaction between 2 carbon compounds, namely succinate and pyruvic acid, acetyl coenzyme and oxaloacetate, fumaric acid and pyruvic acid, malic acid and acetyl coenzyme. Tell. Do you know? So, okay, I'll tell you. So, the very first
Acetyl coenzyme and
oxaloacetate. Good, very correct. So, the first step of Krebs cycle. Okay. Can you please mute your this thing, Mike? I think someone's Kumar Brito. Ha, fine. Thanks. So, during Krebs cycle, the very first step is actually a condensation reaction. Okay. The very first step of Krebs cycle is a condensation reaction. So condensation occurs between acetyl coenzyme and oxaloacetic acid. Okay. So that's why there is a creation of 6 carbon molecules. Now the next question is, identify the correct combination that enhance the activity of Krebs cycle. Okay, I'll just show you. This was week 2. Yeah. Is my screen visible? Is my screen visible to you? Can you see the PPT that has been?
Yes, ma'am.
I just wanted to show you one thing here. Here these are written that these it decreases the Krebs cycle, it increases the Krebs cycle. Now my the only point that I want to tell over here is that there is no need of mugging up, that I have to learn, okay, ATP NADH ATP NADH. No, you have to apply very simple logic. Okay, so what is the ultimate production of Krebs cycle? What Krebs cycle produces? Ultimately, there is generation of ATP that occurs, right? Okay, there is a generation of energy. So Krebs cycle is occurring so that there is energy. So I'm giving you an example. So when you eat food, when you are hungry or when your stomach is completely full, when you have more urge of eating food, not the junk food that you can eat anytime, burger, pizza, no, the good food, the healthy food, the food that is cooked in our homes, when you want to eat that food, when you are hungry or when your stomach is full? Tell me. Basic question. Are you sleeping? Are you still sleeping? I'm
When we
trying
are
to just,
hungry.
when we are hungry, I'm just trying to lighten up of the mood, that's why I'm saying this. Okay, so we try to we consume food, we eat when we feel the urge of it. Okay, when we feel that okay, we are hungry, we have to eat it. When our stomach is already full, then there is no urge that comes out, that comes within to eat. So likewise, If there is already a lot of ATP present, a lot of NADH is present, a lot of FADH is present, then why would our body will use the energy or use the time or the resources to do the Krebs cycle? Because ultimately Krebs cycle is producing what? Krebs cycle is producing ATP. Krebs cycle is producing NADH. But if our body or if the cell already has ample amount of ATP, NADH, then is there any need of more ATP, NADH? No. Then there is no need of Krebs cycle. Again, someone has unmuted the mic. Can you please, Mamita? It creates actually a little problem. Can you just mute yourself, Mamita?
Yes, ma'am.
Yes, Mamita. Can you unmute yourself? Mute yourself. You have already unmuted. Please mute
Okay.
yourself.
I use B.
Sorry.
B. ATP.
I'll tell you, but please mute so that it creates this disruption for other students also. No, that's why I'm saying. Please mute yourself,
Okay.
or yeah. Thank you. Please mute.
Ah.
Okay. Thank you, Mamita. So, I was saying that if the cell already has a lot of ATP, a lot of NADH, then there is no need of more generation of ATP and NADH to Krebs cycle. Is this logic clear what I'm trying to say? Is this logic clear to everybody? This simple logic?
Yes.
Okay. Now you see here. Hmm. You see here that the Krebs cycle is getting decreased, or the activity of Krebs cycle is reduced when there is ATP present, when there is NADH present, and when there is acetyl coenzyme A present. Because pyruvate is converted to acetyl coenzyme A and we get one NADH molecule that we have studied. So if already ATP is present, if already NADH is present, if already acetyl coenzyme A is present, then what is the need of making this whole Krebs cycle? So this Krebs cycle would be reduced. But if there are ADP there are NAD plus, there is just coenzyme A, and fatty acid is an exception, and just you have to learn calcium. So if there is ADP NAD plus, then that means that there is a need of ATP to be synthesized, there is a need of NADH to be synthesized, so that we can give three molecules of ATP. There is a need of acetyl coenzyme A to be synthesized, so that we can give one NADH molecule. So excuse me. So so now we know that the Krebs cycle is decreased when the molecules, that means the Krebs cycle is reduced when the end product of Krebs cycle, that is the ADP NADH, they are already present in ample amount. But if the end products are not present and instead ADP NADH and fatty acids coenzymes are present, then there would be Krebs cycle, so that there can be production of ATP NADH. So now we come to the question number 28, which says that identify the correct combination that enhance the activity of Krebs cycle. Important to note is that here we are talking about enhance. So enhance means the Krebs cycle has to go. So when it will go, when there is lesser amount of the end products. So what would be the answer now? Or is there no, question is right. Tell me what would be the correct answer?
C.
Answer would be, anybody has said something? I couldn't hear it properly. Tell me what would be the correct answer? Nobody is saying anything. Okay. The correct answer would be C. Why? The correct answer would be C Because here the question is tricky because I have made the combinations. ADP will enhance, but NADH will decrease. Fatty acid is neutral, it cannot be. ATP will decrease, NADH will decrease, but calcium ion will enhance. But the pairing is not completely satisfying the question, so it will be wrong. Here NADH will also enhance, coenzyme A will also enhance, ADP will also enhance. So it is the correct answer. ATP no, NADH no, fatty acids neutral. So this cannot be the correct answer. So you have to look for a combination that in which all the three are actually enhancing the activity of Krebs cycle. Here ADP enhances, but NADH does not enhances. So this cannot be the answer. Here ATP does not enhances, NADH does not enhances, but calcium ion enhances. But because two are inhibiting the Krebs cycle, so this cannot be the correct pair. Here all the three are doing. And suppose if sometimes what happens, the question is there that none of the options are correct, then you look for the most correct pair where at least two are matching. Okay, sometimes these kinds of tricky questions also come, then you have to look for a pair which has at least two options which are matching. Okay, let's come to the next question. Which three pathways are connected by the oxaloacetate of the Krebs cycle? Tell me. This is the easiest question, I think, in the whole class. This is the easiest question, and I'll tell you why. But first, tell me the answer. Anybody? Anybody knows the answer? Hmm? No? Nobody wants to try. Okay. Tell Mamita.
Ma'am, can you repeat?
Okay. People are coming now also. Why do we prepare different types of media in genetic engineering? Okay, so Narayan, we will come to that topic and I'll tell you after finishing these questions. When we'll come to the media topic, then I'll explain you. Okay, the question is which three pathways are connected by the oxaloacetate of the Krebs cycle? You even don't know anything. The basic logic is that I have written here three pathways and only A is the option we have actually mentioned the three pathways. So the basic logic can be
Yes,
that
ma'am.
okay, amino acid, nucleotide, and resynthesis of glucose molecules. Now, one topic that you must have studied in your lectures in first class of week 2, that this Krebs cycle is very much important because the products of Krebs cycle, the byproducts of Krebs cycles, the byproducts of the Krebs cycle, they are actually the intermediates, or we can say here intermediates. Intermediate would be a better word, sorry. The the intermediates of the Krebs cycle, they are the intermediates of other pathways also. Okay, so that means कि Krebs cycle is a very important pathway, not because it is producing energy, but because it is linking many other pathways also. So the oxaloacetate, you will see, I'll just show you here. See here. Can you see significance of Krebs cycle? So this oxaloacetate, with the help of the enzyme which is known as pep carboxykinase, it can synthesize glucose. This oxaloacetate, it can also be used for the synthesis of nucleotides like purine and pyrimidine. This oxaloacetate can also be used for the synthesis of amino acids. So that means oxaloacetate of this Krebs cycle is very important. So you can say that this Krebs cycle not only helps in the synthesis of ATP, but it also helps in the synthesis of other biomolecules by connecting other pathways also. It is helpful for the synthesis of glucose, for the synthesis of amino acids, nucleotides, fatty acids, sterols. So that is what the question was. Next question is enzyme required for the conversion of acetaldehyde to ethanol is. So what is the enzyme which is required for the conversion of acetaldehyde to ethanol? Okay. Did you know what is the context? Are people listening here, or you guys are now getting bored? Are you guys listening?
Listen.
Yes, ma'am.
Okay,
C
okay.
Yes,
is
ma'am.
the answer, ma'am.
C is the answer. Okay, answer A. Okay, okay, okay. This Narayan is okay, fine. Yes. So the answer is alcohol dehydrogenase. A quick concept that I wanted to share is that pyruvate, when there is oxygen present, I'll just share it. It will not take much time then. Hmm. So, in the presence of oxygen, you can see here that in the presence of oxygen, the pyruvate it goes to the TCA cycle. But when the oxygen is not present in sufficient quantity, then this pyruvate has two fates. The first fate is that this pyruvate can be converted into lactate, and the second fate is that this pyruvate can be converted into ethanol. Okay, so the conversion of pyruvate to lactate, it requires lactate dehydrogenase. Now, if the enzyme is saying lactate dehydrogenase, how to remember? So you can see that during pyruvate to lactate conversion, there is a removal of hydrogen. That means a hydrogen is removed from pyruvate, and therefore the pyruvate is converted into lactate. Now hydrogen is removed. That means what kind of enzyme will required? Dehydrogenase, which will remove one hydrogen. Okay. So and because it is forming lactate, so lactate dehydrogenase. Then pyruvate is converted into acetaldehyde, and acetaldehyde is converted into ethanol. Now, again here, there is one removal of hydrogen. So, again dehydrogenase will come. Here, why decarboxylase is there? Because carbon dioxide is getting reduced. So, this is very easy. You need not to mug up the enzymes. Like this also you can learn. Okay. Now, next question is, in the absence of oxygen, pyruvate can be converted to, just now we have discussed. What is the answer? In the absence of oxygen, pyruvate can be converted to Fida Shahi. The class starts at 7. Okay, so 7 to 9 is the class timing. So every Tuesday, 7 to 9, we will discuss whatever is been taught in the last week. So this week we are discussing the lectures of week 1 and 2. In the next Tuesday class, we'll discuss the lectures of week 3, and we'll do some questions. Okay, I hope I answered your question. So, what is the answer of this question? In the absence of oxygen, pyruvate can be converted to? Tell me.
Lactate and ethanol.
Yes, lactate and ethanol. Good. So that's just what we have studied. That in the absence, pyruvate can be converted to lactate and ethanol. Okay. Now we come to a little bit easier part. Then again the kinetics is a little bit difficult, so I'll see how much I'll be able to complete the kinetics. If one or two numericals of kinetics are left, we'll do in the upcoming class. Okay, at the first thing we'll do the questions, because those questions are important, and dealing the questions in the last means you all will be completely fed up. Okay. So now we come to the basics of biological system. So now we know that there are different kind of hosts that we can use. We can use prokaryotes, we can use eukaryotes. We know the properties of prokaryotes and eukaryotes, and we have also studied the metabolism that occurs in prokaryotes and eukaryotes. Now we want to culture it. You know what is the meaning of culturing? Growing, propagating. Okay? In these terms we say it propagating. Okay? Um, ok- Narayan, you have any query? You have raised your hand. You have any query? I will answer your question now. That why different medias are needed. I think it's not there. Anyways, so now we see that when we want to culture, when we want to propagate ourselves, what kind of mediums we needed, what are the different types of medias that are needed. So in this particular module, we have studied the basic components of growth media, the different types of medias required for bacterial growth, for yeast growth, then how do we prepare the mediums, and then we come to the mammalian culture media. So the basic growth culture media, what are the basic needs a media needs? Just what the basic requirements of a cell are. So what are the basic requirements of a cell? We need carbon, we need hydrogen, we need oxygen, we need nitrogen. Okay, then what we need? We need vitamins, we need some growth factors, hormones we need. Okay, so these are the basic components of, especially these are the components of a mammalian cell culture media. Okay, now we come to the question. Quickly we'll try to complete our today's lecture. So ratio of medium volume and culture flask during microbial medium preparation is important for, what is the answer? Maintaining the
To
sterility
maintain
for,
the sterility of the medium.
no, that is not the correct answer. Think why am, what can be the logic? This is my culture flask. Okay,
Avoiding carrying the medium components, ma'am.
no, that is also incorrect. Okay,
Proper aeration.
yes, proper aeration is the right answer. Now I will explain you why. So if this is a 1 liter flask, okay, and this is a 250 ml mark, this is not like a practice that is matlab this is not a this is a practice that I do in my experiments. Okay, this is not something which is completely you have to follow this only, but this is what I do is that I take a 1 liter flask and I fill the media up to 250 ml only. Why? Because when you put this media for autoclaving, for sterilization, there has to be some space here so that aeration can occur. Okay, so that there can be some aeration. If I fill my flask up till here, then will there be any space left for proper aeration? No. So that is why the ratio of medium volume and culture flask is important, so that there is proper aeration, and proper aeration is important for the proper mixing and everything. Okay, so whenever you are autoclaving any media, you have to make sure that there is ample amount of space left so that aeration can take place. Now, next question is, which antibiotic is commonly added in mammalian cell culture media? Tell me.
Both B and C.
Yes, both B and C. So basically, I don't know, anybody of you have ever seen at least some mammalian culture, this thing, mammalian culture or any animal cell culture? Have you seen animal cell culture? Has anybody
No,
seen?
ma'am.
Okay, fine. So basically, when we make a mammalian cell culture media, so in the media we add a cocktail of penicillin and streptomycin, and we call it as pen strap. It is a general name, we call it pen strap. So pen strap is basically a cocktail which has penicillin also and streptomycin also. Fine. We'll come to the next question, which is, reason for heat inactivation of serum is degrade growth factors, reduce the activity of hormones, sterilize the serum to remove immunologically active molecules. What is the answer? First of all, do you know what is serum? Do you know what is serum and why it is used and where it is used? Anybody who knows it?
It used in animal cell culture.
Yes, it is used in animal cell culture. So basically, this serum is very important component and it is very expensive also. One bottle, one 500 ml bottle costs around 70,000. It is very expensive and it is very important. Why? Because when you are making a media, this media is used for the culture of mammalian cells. So mammalian cells means it should resonate the kind of environment that occurs in mammalian body, in mouse or in humans. It should resonate, it should reflect. Okay, so we and mammalian cells, it needs growth factors, it needs hormones for the growth. Okay, so this serum actually provides the growth factors and the hormones for the growth of the cell. Is it clear? So serum is basically
Yes, ma'am.
used in mammalian cell culture, which is used to provide the growth factors and hormones. First thing. Second thing is that this serum no, what has happened to my this thing? Okay, let it be. This serum is heat inactivated. Heat inactivated means that before using this serum, we keep it at around 56 degrees Celsius for about 30 minutes. Okay, so we do it so that the immunologically active molecules are removed. This is the answer. The answer is D. What is the meaning? The meaning is that I want to use serum in my culture so that I can get growth factors and hormones from it. I don't want the serum to give me additional antibodies also, because those additional antibodies will hamper my culture. Okay, because serum will not only have growth and hormones, serum will have everything. Serum will have antibodies also, serum will have complement molecules also, but I don't want that. Therefore I will heat and activate it so that I only have growth factors and hormones that I require for my culture. Okay, so we heat and activate serum so that the immunologically active molecules, such as immunologically active molecules means immune molecules. Immune molecule is what? Antibody is an immune molecule. In immunology you study about antibody, right? So that immune molecules are removed.
Yes, ma'am.
Okay, next question is, buffering agent in mammalian cell culture is, what is the buffering agent which is used in mammalian cell culture? FBS, what
Sodium
happens?
bicarbonate.
Yes, sodium bicarbonate is the buffering
Absolutely.
agent. FBS, just now we have studied, it is used for growth factors and hormones. Okay, now we come to the week 2 basics of biological system, lecture 3. This is the last lecture, and we'll very quickly try to come to one or two numericals. They are very easy, but they are extremely important for all your competitive examinations. So in week 2, we have studied about the microbial growth kinetics. First, we have studied about the different types of divisions that occurs in bacteria: binary, budding, fragmentation. Then we have studied about the methods which are used for microbial growth: microscopic count, serial dilution, and all these. And then we have studied the stages of normal growth curve: lag, log, stationary, death phases. Okay, this we have studied. 1 minute, people are still entering. Fine. So the first question is, match the following. So can you tell what would be the matching? Budding is used for budding. What happens in budding? This is a parent cell and there will be a daughter bud, and ultimately it will pinch off. Okay, so budding is daughter bud. Fine. Spore formation. So some bacteria, they form spores during adverse condition so that the climatic conditions can would not affect their bodies. So spore formation is adverse condition. Binary division means, binary division, this is one and it is making like this, it is getting divided. This is a transverse line, and septum is a thing which divides. So binary division would be transverse septum. Fragmentation in filamentous bacteria. So some filamentous bacteria, what happens? So if suppose this is a bacteria and it is this is the filament. So these kinds of fragmentations occurs in filaments. Okay, like this. So this is fragmentation which occurs in filamentous bacteria. Now we come to the questions. Do I have only questions or some okay, first we'll do these questions and then we'll come to the numerical part. Okay, or should we do the numerical part first? What you want? You want to do the numerical part first, or you want to this thing, do these conceptual questions
Numerical.
first? Numericals. Okay.
Conception.
Okay, so I'll go with the sequence, and whatever questions are left, these numerical parts, if you're not able to understand, again in the next class, before starting this thing, the week third, we'll again discuss this question and we can solve even more questions, okay, if you have any problem. So this question says that during serial dilution method, suppose the number of colony observed is in is 200, in it is not in, it is 200 using 1 is to 10,000 dilution. What will be the count of bacterial per ml? What would be the answer? So what we do in serial dilution is that during serial dilutions, whatever number of colonies we are getting, suppose I get 100 colonies, okay, and 100 colonies I got in 1 is to 1000 dilution, then my total number of bacteria would be 1 into 10 to the power 1, 2, 3, 4, 5, 10 to the power 5. Okay. This would be this much of bacteria per ml. This basically is the dilution factor. So we have to multiply the number of bacteria with the dilution factor. Okay. During serial dilution questions. Did you got this? So what would be the answer here? What would be the answer here? Tell. I explained you the concept. Now what would be the answer? Anybody?
200 into 10 raised to 5.
See, here it is written that
Option
number of yes, anybody? Yes, you can say. We have to calculate. To calculate na. Actually, there is nothing to calculate here. But if you want to calculate, you must have a copy or a pen with you. You can just calculate. Anyways, there is nothing to calculate. Okay, I'll show you. Number of bacteria is 200. Okay, and the dilution which is used is 1 into how much? How many zeros are there? 1, 2, 3, 4 zeros are there, na?
4.
10,000 it is. You have to multiply it. So it would be 2 into 10 to the power 1, 2, 3, 4, 5, 6.
2 into
2
10
into
raised to
10 to the
6.
power 6. Or 200 into 10 to the power 1, 2, 3, 4. Anyways, you want to write. So this is not right. This is wrong. This is right. So your answer can be given in this form also and in this form also. So you have to see and then you have to write the select the correct option. Okay. So the main thing is that you have to for serial dilutions, the number of bacteria whatever you are getting into the dilution factor. Okay. This you have to do. Is it fine to everybody? Okay. In turbidity method, blank represents what? So basically, turbidity method is a method for measuring the amount of bacteria that has been produced. Okay. So if it is more turbid, matlab more bacteria. Okay. Are you guys understanding or not?
Yes.
Hmm? So, do you know why we use blank? What is the concept behind using a blank? So basically, suppose I have made my media which is LB, Lurea bertini, okay, and I have cultured my bacteria. And now I want to know how much bacteria has grown using this turbidity method. Okay. So suppose in the spectrophotometer, I have a cuvette and I put and I get a reading. Okay, I get a reading of let's say 0.5. Now, how would I know that this reading of 0.5 is because of the bacterial growth or because of the media? Did you understand? That 0.5 is actually the reading which I am getting not only from the growth of bacteria, but but it includes the reading of the LB also, the medium also. So what we do is that we first run blank. So what we do in blank is that whatever medium we have used for culturing, first we will run this. I will first run LB in the spectrophotometer, okay? And then I will run my sample. So my sample is dissolved in LB. Now bacteria has grown in LB. This is my sample. Okay, suppose this is 1 and this is 2. So my OD should be 2 minus 1, and that would be my OD that it automatically gives. That means the OD, and now whatever OD I am getting, 0.3, whatever I am getting, this OD is only because of the growth of bacteria and not because of the of the media. Because we have subtracted the OD which was coming from the media. Did you understand it? So the use of blank is to normalize it. So normalizing means that we want that whatever results we are getting, whatever OD, whatever reading we are getting, that reading corresponds directly to the growth of bacteria and not to anything else. Not, it should not give the reading of the media. So here the question is, in turbidity method, the blank represents, so blank represents the specific medium which is used for bacterial culture. So in turbidity method, what we'll do, we'll take two tubes. In one tube we will have blank. The blank would be the medium which is used for bacterial culture, and the other tube would be the bacterial culture. So first we will run blank and then we will run our bacterial culture tube. Okay, now next question is, dry weight method is better than wet weight method. Why? Because dry powder is easier to measure. Dried powder can be stored easily. Wet weight has liquid part which can create variations. Dried pellet can be stored for a longer time. What is the answer? Tell me.
Easy to measure.
No. No. Okay. I'll tell you the logic behind it. I have two appendoffs. Okay. These are my two appendoffs. This is a pellet. I have centrifuged and I have tried to remove the media also. I have done my best to remove the media, but still there is some droplets of media left. Okay. This is another centrifuge. Again also same thing I have done. This is the pellet and I have tried my best to remove the media, but still here I couldn't remove much, so there is more droplets are there. Okay. Now I take the weight. I measure it. It will give me a weight of X, and because the media is something more, so it will give me X dash. Okay. The pellet is same, but the weight is different because this this media component is also there, and this media component is also not same from each and every tube. It depends on our handling. Sometimes we are able to remove the complete supernatant, sometimes we are not able to remove the complete supernatant. So wet weight means that there is some media also left, and dry weight means that what I will do, that because I know that there are some water medium, there are some solvent molecules still left, so I will dry it. I will dry and I will make it a powder. Now when I am measuring it, I know I will get the exact reading. Could you understand the point? So, wet weight has liquid part which can create variations from batch to batch. This is the answer. Could you understand this? Tell me if you couldn't understand me. Is it clear? Why? Why dry weight method is better? Because in wet weight method, the liquid part is not completely removed. So this will also give us some reading which is false positive. And also this liquid part is not same in each and every, so that we can minus it. Okay? Now we come to the next question. Huh. I think now we just have one more slide left. Fine. So on comparing the growth curve of a microbe cultured in two different medias A and B. So we have a single kind of microbe which is X. Okay. I have cultured the same microbe in A culture. I have cultured it in B. So these are two different medias I have used. This is A media and this is B medium. Fine. Now, what I have observed is that the bacteria grown in medium A, it has a shorter lag phase. Here, the lag phase is short lag phase. Okay. Here, B may we have a longer lag phase. Fine. Now, what can be the best possible explanation of this? Try this question and tell me the answer. What can be the best possible explanation for this? Hmm? Try. I think I have to stop at 41 only. These three questions calculate the number of bacteria we'll do in next class. Will that be fine to you? To you guys?
Okay, ma'am.
Because
Okay,
Okay,
ma'am.
ma'am.
because these three questions are actually important and I want your full concentration. And we don't have that much time also. So first tell me the answer of this question. Question number 40. What do you think would be the answer of this question?
Option D.
Option
D.
B or D? What you are saying?
A said the
I
bacteria
could,
to
okay,
adapt.
you're saying D. Yes. So option D is the correct one. So in the in the subsequent online class, I'll in detail I will explain you the lag log phase. But here what I'm trying to say, I'll just brief it up. So see that bacterial growth has bacterial growth kinetics. Again, bacterial growth kinetics is very important, children. This kinetics will you will be experiencing in biochemistry also, and you will have this in bioprocess or this thing microbiology also. So this is very important. So bacterial growth kinetics, if you see, okay, this is the lag phase. This is the lag phase. This is the log phase. This is the ex sorry stationary phase. This is the stationary phase, and this is the death or decline phase. So what happens, that lag phase means that suppose there is a new suppose you study in a school and your father got transferred and now you go to a different school. Okay, so on your first day or second day or for a week, you will try to adjust with the new environment. Okay, like when you first came to the university or college for your graduation, you took some time to make friends, you were knowing, you were adjusting, you were seeing the atmosphere, and after some time you get acclimatized and then you have very good friends and then you enjoy a lot. This is what happens with us now. We take some time to get used to the environment, we take some time to get adapted to the environment. Likewise it happens with the bacteria also. So when you put bacteria into a medium for the growth, so bacteria will take some time to adapt with the environment. Okay, so this adaptation phase is known as lag phase. Now, if you are in a university and in the first day of college you got a very good teacher who motivated you a lot, then you become very motivated, very enthusiastic, and it took lesser time for you to get adapted to the environment. But on the very first day, you some teacher came that scolded you off, and he said that you won't be able to do anything and demotivated you a lot. Then you will become very sad, you will become very depressed, and you will take more time to get adapt to the new environment. Okay, so same thing happens with the bacteria also. So if the medium is very good, that means medium has all the components that are needed for the bacterial growth, all the growth factors, all the hormones, every not hormones, not growth factors, we're talking about bacteria. But if the medium has each and every component which is required for bacterial growth, bacteria will take less time to adapt and therefore the lag phase would be short. Okay, but if the medium is not having all the components for its growth, then media will take long time to adapt and therefore the lag phase would be long. Could you understand the logic what I'm trying to say? Yes or no? Tell me.
Yes, ma'am.
Okay. So, yes, to Mamita, you have any query you can ask. And so, here the question is that when a bacteria X, it was grown in two mediums, medium A and medium B. So medium A, it has a shorter lag phase, and medium B, it has a longer lag phase. So what would be the best possible explanation? The components of medium A, it supported the growth of bacteria, and therefore the bacteria took lesser time to adapt. Whereas the components of B, it kind of create a stressed environment. That is why it took a longer time to adapt. So what we can get is that when a bacteria is taking lesser time for adapt, then it will have a shorter lag phase. When a bacteria will take long time to adapt, then it will have a long lag phase. Okay. Now see, here I have written the best possible explanation. Now the reason can be this also, that because medium A was in more quantity, so more quantity means more factors, so more factors means more growth, or the it can be anything. But see here the point is to know the best, to know the exact possible explanation. The exact possible explanation, the logic is that medium A has supported the growth. By what means, that is secondary. The first thing is that it has supported the growth. Okay. Now we come to the next question, which would be the last question of today's class, and then we'll dismiss our today's class. So the question is, when the substrate is limiting, the bacteria go in which phase? Tell me the answer. Last question, come on, try to answer the question. Tell me, anybody who wants to
Option
try?
B. Option B.
No, option B is wrong. So
Ma'am,
we know this.
remain in the lag phase, ma'am.
D, ma'am. Remain in the lag phase.
No,
D.
I'm
no.
stationary phase.
Now we know what would be the answer. Anyways, so uh, for this substrate limiting, I will explain the logic behind it in the next class, because it will take a lot of time. But before that, I want you all, this whole recording would be shared on my YouTube channel. Uh, I'll share the link of the channel. I In the next class. Now I don't have the channel yet. Anyways, so I want you people to learn about to read this growth kinetics again, and obviously you will have some other topics that will be covered in the next week also to read, but at least you can just go through the kinetics once more, so that it would be easier for you to answer the questions. Okay. Because this is the most important part. The two very important parts of today's lecture, first was this metabolism, which is very important because questions are asked if you are preparing for any higher studies or any competitive examinations like CSIR or GATE. That is very important. And the second part, which is very very important, is microbial growth kinetics. Each and everything is important, but this is like a tricky one. So if you are taking an online course, you are taking out time from your regular classes, then you should try to get the best knowledge that you can get from here. Okay. So I am also a student, so there would be a lot of things that I might say wrong, or even I might not know, or I will learn from you. So we all are students here. So but my whole attempt is that I will try to give you the best, the knowledge that I have, or that experience that I have in solving these questions, and experimentally also. So apart from that, you can always ask me questions after the class, like after the 2 hours. If you want, we can, after this first session, you can always uh, ask me if you have any doubts of what kind of colleges you want to apply further, and what courses you want to pursue, whether you want to go for higher education, what would be the good colleges. I can help you in that also. Fine. So we'll end today's class now. So we'll meet next Tuesday, and hope you all would be there, and please try to just read about this growth kinetics. Is lecture material sufficient for final exam? I think so. It would be sufficient, although the exam paper would be made by sir. The important thing is that you should know the logic and concepts. If you know the concepts, you would be able to solve the questions. Plus, you have to submit your assignments also. So, as per the rule, we cannot discuss the assignments here, but I think I have covered almost all the topics that are there in the assignment questions. Okay, so I think it would be if you are attending the online classes plus the offline courses, I think you will be able. For some question, we are not able to find answers in materials. How can we reach out to you? Okay, so I am writing my mail ID here. So if you have any queries, you can, where can we find? Please suggest. You want some material. Okay, so Narayan, so which course you are in? Like you are a BTech, BSc, MTech, MSc. In which course you are? Undergraduate, postgraduate level? MSc you are. Okay. So you do you have Pathfinder? See, Pathfinder is not a very good book. Like good as in that it is good for last minute studies. It is okay. It is always best to go with the proper reference books. Leringer, Stryer, or for microbiology there is Prescott is there, which is very good. For this thing, for molecular biology, you have genes. Genome is also there. But if you want like
Ma'am,
very, huh?
could you please suggest a good book for molecular biology, cell biology, and genetics? Because I have all these three subjects for my course, and our teacher suggested few books, but still, if any more.
What books you are reading? Do you read Alberts for cell biology? Have you read?
No, my teacher suggested us the Verma Agarwal book, which has both cell biology, molecular, and genetics, and the environmental studies.
Verma Agrawal. Okay. So, with due respect to your teacher, I think it would always be better, especially for our subject, to read some very famous foreign author books, because they are like they are the main sources from where people actually make the material. So you can read Alberts for cell biology. Okay, you can read that.
Okay.
And for molecular biology, you can study genes. There are many editions of it. And for microbiology, you said
No, no, genetics.
genetics. I think these two books will have sufficient material for you that can be covered. You can also read Prescott. The spelling may be wrong, you can check it. Or Pelzar for this kinetics. uh, the spelling may be wrong, you can just check. I'm not confirm about the spellings, but yeah, these books you can read. And yes, I think any other questions do you have?